This is going to feel rather different from the morning session. So Varun will be telling us about evolution from a machine learning perspective, as an analogy with how learning can happen versus how DNA seems to learn how to respond to the environment. Varun is one of the world experts in this new area of trying to understand evolution with this analogy. So very happy to have him here.
Well, thanks, Paul. Again, so please interrupt me if there's anything which I say which is not perfectly clear, and there might be a fair number of subsets. So before I start talking about evolution, let me even begin with machine learning. And there's no shortage of applications where machine learning has been extremely successful, right? So things like speech processing, object recognition, language translation, and there's any number of more applications in which machine learning has been extremely successful. And what we find is at a lower level, all of these things, which might look as very different tasks, have very similar or almost essentially the same algorithmic techniques. Okay, there's the same algorithm that can be remarkably successful in any number of tasks which look very different from each other. And it seems like evolution is also actually rather successful at doing remarkably different things. And the question is, can we use sort of what we understand in learning to have a different look at evolution, right? So for us, let's say evolution by natural selection starts with Darwin and Wallace, and enough has been said of this. I won't say anything more on that. But what I do want to talk about in this talk is going to be, I hope, significantly different from what has been said here so far. And if you look at living organisms, they have complex mechanisms. And what do I mean here by complex mechanisms? In particular, they operate very differently in different environmental conditions. So they're actually doing something that's they sense their environment, and they're doing different things in different settings. Okay, and the question, of course, what we need to know is what we want to answer is, how does this complexity arise? And random search, maybe in the genome space or whatever it is, cannot alone explain for all the complexities as we observe around us. So there just hasn't been enough time, and there aren't enough resources for random search alone to explain why evolution seems to have been so successful as we think it is. Right, but of course, Darwin's theory says that there's random variation acted upon by natural selection, and what we're interested in is, can we quantify the notion of complexity that can arise through through the process of random variation and natural selection. Okay, and what's the notion of complexity that we want to use? So tomorrow, I think Daniel and Ian will talk about evolution of complexity in maybe some other ways. But for now, what I mean by complexity is, if we look at organisms or cells or whatever that's computing functions, there's a mathematical function that's being realized, and that's going to capture the notion of complexity for us. And hopefully things will be clearer as we go along. Okay, so let me look at the picture and look at a particular function that's going on in the cell. So here we have a cell, and think of a particular function, say expression levels of proteins. Right, so we have a cell. There's all kinds of things on the cell wall that are sensing things around it, maybe even inside the cell. So there's all kinds of sensors that are sensing the environment. Right? And based on what's going on around it, the cell has to decide whether a protein should be produced or not, or how much of it should be produced, and so on. Right? So if you think of it as a mathematical function, you can write the quantity of protein that's produced as a function of any number of variables. So for example, this could be, think of each of xi's as representing concentration levels of other molecules, macro or micromolecules, that are present around the cell, and that's some combination of that should determine whether or not a protein should be produced and how much of it should be produced. So this is a function that's being computed in the cell, and this is happening all the time in every living organism. Right? And the question is, what are these functions, how complex are they, and how does evolution succeed in finding them? So these are the kinds of questions we want to think about. Right. So before I go on, so I want to talk about the notion of an ideal behavior, and this could be a hypothetical entity. So think of the ideal behavior. So it would be helpful to maybe think of this example whenever you want to think about what I'm saying here. So think of ideal behavior as optimal expression level for a protein for every possible environmental condition. So an environmental condition is represented by some numbers x1 to xn here, and for each of these settings there might be some ideal amount of protein that should be produced, which could be 0 to anything else. Okay, and now we also want to know. Yeah.
It's a different optimal level depending on the conditions. Is that
It's a different optimal level depending on the conditions. So the simplest form could be either 0 or in sufficient quantities to catalyze some reactions or something, but you could imagine something else. And so the measure of performance is going to be how close is the actual function that the cell is computing. So the cell is computing some function which is very different from the ideal. So how close it is to the ideal. And so what's the notion of close? Well, it depends on what kind of values these x1 to xn can take and with what frequency. So think of there's being some distribution over any possible values of these variables that can be encountered in the lifetime of a cell. And on average, the cell has to be as close to the ideal as possible to be considered better than others that are not. So this is the level of performance of under a particular distribution over these possible environmental conditions. How does the actual function computed by a particular cell compare to the ideal function?
averaging over that distribution or averaging the exponential?
Averaging over the distribution. So there's a distribution over, think of these as distribution over the x-size, and I just want to, I have some notion of, yeah. Okay. And so this performance in some sense defines a fitness landscape for every different kind of function that can be expressed by the cell. There's a different performance, and the ones that have higher performance are more fit than the others. Okay, so this is. Okay, and the question that's of most interest here is, so what kinds of functions are evolvable? This is the question we want to try and begin to answer. And the first constraints that we of course need to impose is that we know that you need to be able to do this in reasonable population sizes, because evolution doesn't have access to exponentially large population sizes, as Christos mentioned this morning, and also the number of generations. So there's been, say, maybe 10 to the 12, 10 to the 14, or some number of generations. There's a fixed. We've had only so many generations, and these functions can we explain how the functions that we might observe arose in these many number of generations? So these are the constraints that we have in this evolution. And so I want to emphasize again, so in this talk, what we're talking about is the evolution of genes themselves, right? So the functions of the cell compute is encoded in the DNA. That's a gene somewhere, and that itself is changing. So mutations are essential to understanding any of this. And so we're not talking about population dynamics or just how allele frequencies change in the population, or so these kinds of questions. So they've been discussed in many of the previous talks. So the question what we are concerned with is how does complex novelty arise through the process of evolution, and can we quantify this? Okay. Yeah.
Let me just clarify from a biological point of view. When you're saying how does complex novelty arise, what do you mean by novelty first? And second, are you okay? First, why don't you tell me what novelty is for you?
Right. So, okay. So at some point of time, you had cells that were not showing this kind of behavior. And some cells started showing this behavior that controlled levels of certain proteins, and that gave them selective advantage, right? So that's a novelty. So some organisms learned that it's actually important to produce certain things only in certain conditions, as opposed to sort of always having these levels.
So you mean a new cellular phenotype?
A new cellular phenotype. So these functions, are essentially phenotypes, yeah.
They really from regulation.
From regulation, yeah.
And so your organism is a unicellular organism. You're looking at complexity at the level of the cell here for your practice.
So for now, it would be fine to think of it that way. I mean, this theory is not restricted to that, but it might be the simplest way to think of, yeah.
And so should we think of your function f as being parameterized by the genes?
By the genes. Okay, so the function itself is encoded in the gene. Okay, so the gene represents f. F. Yeah, so it's encoded in the DNA. Right, so
it's this one F?
so of course there is multiple Fs that are going on in any living organism, but if we're going to focus on just one F, so if selection is acting only on a single phenotypic behavior, can we explain what complexity can arise in this?
But you're still going to look at many possible conditions. So
We're still going to look at many possible conditions.
So it's still the
Right.
fitness is a complicated thing. It's not just
Fitness is a complicated thing. It's only the single phenotype that we're talking about, single phenotype, not multiple ones.
And what's the range of f that maps to Booleans or
Okay,
high-dimensional?
Yeah. Okay. So I'll talk about that. So for the most part, I'll focus on f being a Boolean function, but you can of course let f be real valued and high-dimensional real values. Actually, Paul has some very nice work in that, and I'll mention all possible. So for right now, you can just think of the output being in some abstract space, but most of my talk will be about Boolean valued functions. So maybe.
So, one way of looking at much of this is you've got your favorite phenomenon that's happening in some organism, in some cell or something. This organism responds to some environment in some unusual way. So how did this evolve? Could it have evolved in one step, or did it need to have evolved in multiple small steps? Did it need to have been trained? Was it fortuitous that it arose, or will this kind of thing always predictably, easily evolve? So can we start understanding the classes of behaviors that can easily evolve versus the class of behaviors that are sort of surprising?
Thanks. That was the perfect thing that should be said at this point of time. Right. So now I'm going to describe in some detail a computational framework for trying to understand this question. Right. And this is due to Les Valiant. Oops. Around 2006. Right, so it'll be helpful again to have just this picture in mind.
I'm sure you know how to spell this.
Yes. Okay, so we're going to let say X be a set that represents environmental conditions. And typically what we think of this as some vector in high dimensions. So these Xi's could either be Boolean or real valued. So there's some high dimensional vector that represents environmental conditions. Okay. Think of it as those concentrations there that I have, and there's distribution d over x, which is the one that's going to be used to compute the performance, which is how likely a particular environment is. And what we want to answer is, can we characterize for what ideal functions do we expect evolution to succeed, and for what maybe we can argue that evolution cannot possibly succeed in any reasonable amount of resources. Right, so the ideal functions f, we'll think of them as coming from a class of functions. Let me call it f. Right. And so what are some examples of ideal functions? So simple example, if everything is Boolean here, would be conjunctions, which says that f of x1 to xn is x1 and x7 and x13. So it can be, so it's a conjunction, but on some subset of the variables, you don't know which ones. And what you want is that the function should be 1 if this holds, otherwise it should be minus 1. Right? So think of it as, over here you have, if some concentration levels of these particular things are high, you should produce the protein, or otherwise you shouldn't produce the protein. So this is one possible ideal function. Consider.
It's kind of a strange one in a sense, because you're just saying like you hardly ever want to have it on. The more of these,
Sorry?
like the more of these conditions you add, the less often it should be on, right? I mean, like, so it's not like it's a very tricky rule to figure out whether it's on or off. Just turn it off all the time, you get pretty close.
So it depends on how long this is. I mean, if there's only 3 or 4 of them, that's not
Yeah,
true.
yeah, sure.
It also depends on what the distribution over these x's
Yeah,
are. So it doesn't
yeah.
have to be a uniform distribution.
Okay.
So. Maybe I'll just write 1 or 2 more. So if you allow f to be real valued, it's sort of Boolean, we could also consider functions that look like some linear combination. Right? So it's some 7x3 plus 4 minus 13x minus so on. So there's different classes of functions, and some are more complex than others, and you want to try and characterize for which classes you expect evolution to be able to succeed in actually reaching some such representation, and for others where you cannot. Can you argue that you cannot for some other classes of functions? Okay, so now let me define how what an organism is going to be in this setup. So I should write down there. Let's restate the definition of performance as well. If r from x to say minus 1, 1 is the function that's actually computed by an organism, and I'll come to how we're going to represent an organism in a second, then the performance of R with respect to f and d, let's say f is the ideal function, is simply the correlation between f and R. Okay. And if f and R are Boolean functions, then this is, in some sense, this is the only performance metric that makes sense. If there's real valued functions, there might be several other measures that make sense, and we can talk about that later. Okay, so how do we represent an organism? So this is going to be some. So what we're going to say is that an organism is simply a string. So there's a class of strings that can represent an organism. So think of this as just strings over zeros and ones, but they also encode a function. Right? So think of this as a description of a function. So R in R, say if I write it in brackets, it means that it's actually a string. This string that encodes a function from say x minus 1. Okay, and what we want to impose on this function is the fact that this can be evaluated in polynomial time. And what I mean by that is if there's a computer program that given say r and x as input outputs r of x, and this program runs in polynomial time. Yeah. Yes.
So we have a finite number of variables and it's a Boolean function. I thought, I mean, how could it not be? I mean, I thought all such functions could be expressed as polynomials. In the variables, any input variables.
Say that one more time. So there's a. So I can define functions. I can define arbitrary functions that are not computable in polynomial time, right? So the representation of the function. Okay, so the representation itself has to be small. It has to be polynomial in n.
Yeah.
And then there's So there's no reason why all Boolean functions can be evaluated in polynomial time, right? So let's say my x encodes a graph, and my function is say if it's 1 if it has a Hamiltonian cycle or not, or something. It's a function on some input, but it's not. It cannot be evaluated in polynomial time. Okay, and what I want to also let me state at this point what we think of as sort of the goal of evolution. So there's some error parameter epsilon, and what we want to understand is can evolution reach some r such that the performance of r with respect to the ideal function is at least 1 minus epsilon.
Are you going to evolve the algorithm for evaluating the string in polynomial time?
No. So this algorithm is fixed. So think of once the representation class is fixed, this algorithm is fixed. So we're only going to change r. So that's right.
And the question there, if I wanted to switch that to like given a starting string, like how well I can improve it, is that closely connected, or like instead of if I drop the reference to the I say that there's some optimum that's very good, I don't care too much about that, but I say I start somewhere, I want to know how much improvement I can reasonably get. So maybe I can't reach very close to the optimum.
Yeah. Okay. Yeah. Okay. So what you're asking is, instead of this 1 minus epsilon, can we guarantee? Okay. So that's a perfectly valid question. So maybe you think that evolution doesn't quite reach the optimum, and we want to guarantee that. Can you always guarantee progress? How close do you expect these? So you can ask. Yeah.
What access do you have to F? It's just you can only.
So I'm going to say that in a second. So yeah. So it's going to be through natural selection, and I'll describe how that is modeled in a second. Okay. So.
Just to. F is what you call the ideal function,
Yes.
and R is what the organism does.
R is what the organism does.
And this performance is basically the measure of how close they are.
Performance is the measure of how close they are.
But do I know my performance?
No, not necessarily. So everything is only through natural selection. So that's,
Don't make this about you. Yeah.
That's a question of population size. Is it wider?
okay. So the next thing I'm going to define is what mutations can happen to R, and so the mutation process is also going to be defined to be just an algorithm. Okay, so this is an algorithm takes as input the current representation R, and maybe this error parameter epsilon, and it outputs a neighborhood, which is a number of other representations. Right? So each of these Ri's is an R. And what do we want? Well, first of all, we want that M is bounded by some polynomial in n(1/epsilon), and that this algorithm itself runs in polynomial time.
So it's asexual.
It's asexual. Yeah. Okay, and so this is going to be the definition of mutation. So we don't want to get into the business of trying to explicitly model what this algorithm is that evaluates R of x and how a particular function is actually encoded and what mutations can be. But we don't want to let nature have the power to somehow do computations that cannot be simulated in polynomial time and get mutations out of this. Okay, so think of this as allowing as generous resources as you possibly can for a mutation mechanism. Now coming to Costas' question, we also have to say what selection is, because so far there's no reason why this has any access to what the ideal function is. And that will come through selection. And I have the definition of a performance up there. What I'll also consider is what empirical performance. And what this means is simply that this is the performance evaluated in the sample. It's the expectation of xi, f of xi, xi going from one to the where x1 through xm are drawn from this distribution, xs are drawn from this distribution d over this distribution over the possible environmental conditions. And the reason for doing this is because we don't want evolution to somehow act on extremely tiny differences in performance. So it has to be robust enough.
Actually, so back when you were doing the mutation, was there some measure of how the RIs are close to R, or that's just the definition?
No, so this is the definition. So
there's not some inequality that they satisfy?
there's no such requirement. Right? But there's a fixed algorithm that no matter what R I give it, it outputs this mutation. So it's not that for okay,
So in some sense, this is a fairly permissive model that you can have mutations that go really far from your parent's DNA. So it's up to you guys, the audience, to figure out how to interpret this.
Yes.
So if it's a permissive model, then impossibility results should be especially convincing. And so if you can't do it even in this model, where you're allowed to do really big mutations, then that should be strong evidence that evolution can't work this way.
I mean, a big enough mutation could end up being a recombination.
Okay, so sure. So your mutation algorithm can support recombinations, and you can also simulate sexual reproduction as part of the mutation algorithm. So conversely, if there's a positive result saying, well, we can do this under this model, then you, the audience, can ask, well, okay, so is a subset of the model that Varun is taking advantage of kind of small enough that I view this as being reasonable? So some of the capabilities here seem a bit extreme. So are they being used reasonably in the positive results? So this is something for you guys to evaluate as the talk goes on.
What do you mean that it can simulate sex? Because it doesn't depend on the rest of the population. It's just a function of...
That's more complicated.
Yes, it's a bit more complicated, but I'm happy to talk to you after the talk.
Just as far as existence and what the goal. So, is some part of the argument going to be that there exists these algorithms for the string and for the mutation algorithm such that things can happen? Or is it that under relatively general class of those, that certain things can or can't happen?
Right, so that's what I guess Paul was hinting at. So we want to have both kinds of results. Right? So we want to rule out certain classes of functions, and we think even with this, you don't expect evolution to succeed in getting anywhere close to ideal for those function classes. On the other hand, you can show that for some classes you can achieve this, and then it's a very valid question: are these the mutation algorithm that we define, and these are are they something that you would ever expect to observe in nature, and what does that tell us about this? Right, so. Okay, so just I'm going to denote this by say performance hat, and so the way selection works is the following. So for each of the possible mutations and for R itself, we have this approximate performance. And then we can define which among them are beneficial, which is so all Ri such that performance of I is greater than performance of R. And I'm going to put a tolerance factor here. So it's not just that it's better, but it's somewhat noticeably better. And the neutral mutations are those for which the difference between performances is basically within this tolerance, and the rest are deleterious. Right? Okay, so this is.
So why do you need to go to this performance?
Could you repeat the question?
Yeah. So the question is that why do we need to go to empirical performances as opposed to performances? It's also. So one thing is this. So certainly you don't want sort of very small differences in the performance to affect evolution in any way. So if it's infinitesimally smaller than this, then you don't necessarily want to let evolution act on it. So there's some noise inherent in the system, right? So even with this tolerance, it's the process of which mutation is beneficial and which is not. It should not be defined by arbitrarily small performance differences.
But the organism only gets to sample some finite
Yeah,
amount of
yeah, it gets only so it's not, and it doesn't actually know this.
biologically
The value has. Yes, so,
In the empirical performance formula, what is this E that's sitting?
oh, sorry.
There's no E.
This is this.
expression.
Yeah. Sorry.
And
Yeah.
for the performance evaluation of the different RIs, do you throw a bunch of sample once, or you throw for each of them these guys different samples?
So it turns out that some of these questions don't matter as much. So you can show robustness results that you can define them in several different ways, and all of them lead to the same notion of evolvability. But for now, think of it as each state when you're considering one mutation selection step, you draw a fixed sample and then you evaluate all of these representations on the same sample.
But that robustness doesn't come from like some concentration that would allow me to as well replace all these empirical things with the exacting, right? So
Okay, so the robustness, so a lot of the robustness results can be proved by this reduction that Vitali has from learning algorithms to evolution, and then because sort of we know that evolution can be simulated by a certain class of learning algorithms, and all of them can be put in this framework, then you get that basically they define the same class of functions that can be evolved. But it's the answer is yes, but yeah. So, okay. Okay, and so what selection does is now is not right there, but I don't want to erase this either. So let me get rid of this. So the selection rule is if the set of beneficial mutations is not empty, R prime in at random, and if this is empty and the neutral mutations is not empty, pick R prime in the neutral mutations at random. And this is, so R prime is going to be, R prime survives to the next generation, is good to say. Okay. And like the question of robustness here, there's some robustness that you can also ask about selection rules. For example, you can ask what happens if you let evolution select the one that has the best performance among all of these and things like that. So there's a few more subtle points there, but by and large, a lot of those selection rules give rise to the same notion of
This is, I guess I'm confused whether this is an individual and you're keeping a lot of diversity in the population, or is this a sweep that goes through the population, and so after this step you only have one?
So after this step, you have only one. So this generation, what I've written here, need not be one biological generation, but
Right.
this
So
is what ultimately survives. This
Successive
could be-
sweep, where something,
Yeah.
mutation comes in, takes
Exactly.
over,
Exactly.
mutation comes in, takes over.
Exactly. Yes. Okay.
Which is a very restrictive condition. It's essentially small populations.
Restrictive meaning.
Meaning things that, you've got a factor of 10 to the 30th better than you can do. Right? I mean, there's a very large number of individuals, and so things that can't happen here.
But all of this is a polynomial time
Yeah, yeah,
restriction. So the question is, can you or can you not do
Yeah.
this in polynomial time? So factors of even 10 to the 30 are still constant. And
Yeah, okay, yeah.
So you need to put on your theoretical computer science hat for this talk. 10 to the 30 is a constant.
But since you already raised a few minutes ago the issue about which things we should take away, then it's a fair question.
Yeah. So I mean, there are other results about sort of trade-offs between if you add more population to this model, how much does it let you reduce the number of generations?
Okay. So the last thing I'm going to need to define in this whole setup is now what we actually do mean by evolvability. And right, so one step of evolution is with this mutation plus selection. Maybe you start with a representation R, you have a bunch of possible mutations, and one of them is selected for the next generation through the selection rule. And I'll try to be fairly formal in actually writing the definition. So bear with me for that. So we're going to say is that we say a class of functions as a function, say f, is evolvable with respect to a fixed distribution d over x, let's say, if there exists a representation class R a mutation algorithm these sized functions and tolerance, I didn't talk much about them, but the requirement on S is that it has to be at most a polynomial in n and 1 over epsilon, and the t should be at least an inverse polynomial in n and 1 over epsilon.
So what is the representation class?
So unfortunately I erased that, but this was so each representation is just a string, and it also encodes a function. And
Okay,
the
so
requirement
string
is that it can be computed in polynomial form. Yeah. Such that, and so this is things for every f in f, every epsilon greater than 0, and for every say r0 in R, the sequence R0, R1, G, where each Ri is obtained from Ri minus 1 through this process, is such that with high probability, the performance of Rg is at least 1 minus epsilon, and G is bounded by some polynomial in n and 1 over epsilon. Okay, so let you parse this for a couple of seconds, please. Right. But the crucial points to point out here, and so the requirement is that, this is, once we fix a class of functions, a class of representations, and a mutation process, and no matter what the ideal function is from this class, and for any error parameter epsilon, and for any starting representation in this class that you've picked, if you allow evolution to proceed as according to this mutation algorithm and selection rule, eventually you will reach a representation which has high performance in a reasonable number of generations. Yes.
So this condition that it's for all possible f's is quite strong. So are there other notions that for particular classes of
So that means you're restricting the class of functions. So,
yeah.
right, so once you fix your class of functions, you want to talk about whether that is evolvable. This should work for every function in that class. So for example, I had conjunctions up here, so for all possible conjunctions this should succeed. If you could restrict the class, you could say only for maybe short conjunctions, and that's a perfectly valid statement, but then you would have to make the statement that short conjunctions are evolvable.
It's restricted anyway.
Yeah. So, but once you fix your class, you should be able to prove this kind of result as well. Okay, so that was a lot of definitions. So I'm going to try to draw one particular class of functions and give an evolutionary algorithm for that. So hopefully some of this makes a lot more sense. And then in the second talks after the break, I'll actually show that there's a class of functions that provably cannot be evolved. And okay. So, okay, so this class of functions is selection. So this is going to be evolving simple linear threshold functions. So let me fix x to be so x is just Rn, and the distribution is, let's say, is a product normal distribution in Rn. So it's a spherical Gaussian distribution over Rn. Right. And your class of functions f is the form, the f is sine of w dot x, where w is a vector in Rn, norm of w is 1. Okay, so what are these functions if, let's say this is space? These are essentially half planes that pass through the origin. So this is my class of functions. So all of these are, say, +1, all of these are -1. And I could choose any direction and choose a half plane in that direction. So this is the class of functions that I'm looking at.
Each one is classified characterized by a single omega.
Each one is characterized by a single omega, yes. So now I need to define a representation class. And so the representation class is actually going to be f itself. So this is, I'm going to use, again, each representation is going to be characterized by this vector omega, which defines some direction which may be different from here. Now let me define the mutations.
So I guess I don't understand what you mean by representation being a function.
So I'm going to represent, so my R that represented the function that the organism was computing is from the same class as this. So each W defines what the function the organism is computing. Right, so and which is this function. So think of each. Okay. So each R is some W1 through Wn, and this computes a function which is exactly R of x, a sine of this. So the picture in 2 dimensions is a bit simpler, so let me just draw that. So what we're saying now here is that suppose there's some ideal function. So there's some ideal function from this class which is, say, represented by this red vector. So this is f star, and you start with some arbitrary representation w in this space, and you want to give a mutation algorithm that changes to this vector w will eventually get you arbitrarily close to f star. So that's what we're doing. So what's the mutation algorithm? And I'm going to leave some of this as an exercise maybe. So let's start with w. So this takes, then put the current representation r, which is represented by w and epsilon. So let w u1 through un minus 1 form an orthonormal basis for Rn under D. Yeah. And so the neighborhood of W, let's say, is simply going to be W cos theta plus or minus ui sine theta for I mod to n. It's 1 for theta chosen appropriately. Okay, so before I say anything more on that, so what are we saying here really? So let's say this was your ideal function f star. Maybe you started with w somewhere here. What we're going to make changes to w is by allowing it to move slightly in any direction around it. So no large jumps, but some cone around w where it can move in any way. And one of them is going to be in a direction that's going to be noticeably closer to the ideal function than what w originally was.
Theta is a constant here.
No, it depends on epsilon and n.
It's a constant.
Yeah, it's fixed. Yeah, so once epsilon are fixed, it's fixed.
And for the neighborhood definition, can I choose any orthonormal basis? So no W.
So w has to be one of them, and then you can choose any orthonormal basis.
So any orthonormal basis will give me a neighbor.
Any orthonormal basis will give you some neighbor that's actually going to have significantly better performance than w. That's what.
And what is orthonormal basis under the distribution? What do you mean by that?
So they are orthogonal with respect to this measure on Rn.
I see.
Yeah.
Wait, I mean, this would just be orthogonal in our normal.
Yeah. Okay. So because it's a normal distribution,
Yeah,
okay,
yeah,
it's
it
just
doesn't
it's just
change.
orthogonal in a normal sense. Yeah.
Yeah. It's a normal basis.
Yeah, even standard basis would. But I think w has to be one of them, and then no. Yeah. Okay, yeah. Maybe. Yeah. Okay. Maybe even. Yeah. Okay. So yeah. Even the standard basis should I think work. Yeah. Okay. So okay. So I don't want to actually do the calculations, but if anyone is interested, you can.
So wait,
do
is
this.
the UIs fixed for each step, or is it, you can choose it again each step?
So they can be chosen differently. It doesn't matter. So you can fix UI as I think Vitali said as a standard basis ahead of time, or you have W and then you have n-1 more dimensions. Just choose any orthonormal basis in the remaining dimensions that you like, and So any of these things will work. And so one of the advantages here is that this is fairly robust to the kind of mutations that you use. And hopefully at the end of the next talk, I would like to have some discussion on what kinds of mutation algorithms are more reasonable than others or not, and is there any intelligent way to say why some mutation algorithms are better than others or not?
And random vectors chosen.
Random vectors chosen will be fine.
Gaussian don't say that.
Yeah.
Then you need many dimensions. Then you need many dimensions.
Just for about n random vectors.
Yeah. Yeah. Yeah, and sort of the main thing to use here is that would be helpful is that the performance of this is basically related to the angle between W and F star. So your performance is nothing but the angle over, say, is the performance of W, simply I think theta over 1 minus theta over. So it's because it's so W classifies all of these as positive, F classifies all of these as positive, and what you have is anything that lies in between is where W and F disagree. And so the performance is just related to the angle between them, and using elementary trigonometric inequalities gives you a fact that these kind of mutations will actually succeed in.
It's not just that it's a one-dimensional function, it's also that it's a very nicely behaved one. If you made it like w dot x bigger than a third.
Yeah. So for this particular thing, it's critical that you're looking at thresholds that pass through the origin.
But does it depend on the tolerance? Because we might expect that if the weakly deleterious ones that you allowed too many of those, that the neutral mutations coming in would drive you away from them.
So it does depend on tolerance. So you need to tune the tolerance correctly. But these kinds of mutation algorithms are actually such that if you choose your tolerance correctly, you can actually guarantee that there's always a beneficial mutation, and that's not always the case. But in this case, it is, and which means that you can set the tolerance to a reasonable inverse polynomial, and you will get that this succeeds in actually a reasonably small number of generations.
But your mutation algorithm, you pick a given number of possible mutations in this neighborhood.
Yes.
So there's some parameter which is how many
Yeah.
of those you choose.
Yes.
Which you haven't specified.
So, I'll take any more questions, but otherwise this will be a good time to stop. And then after the break, I'll talk a bit more about learning theory and then show how that can be used to show that there are actually functions that cannot be evolved in this cellular.
Let's say